Hi, hello guys, welcome to my channel. I am Vishnu in College in Tirupattur. In this video, I talk about heterocyst and its importance in nitrogen fixation. And first of all, introduction, then identifications of heterocyst, definition, heterocyst type, positions of heterocyst, structure of heterocyst, development of heterocyst, factors affecting development of heterocyst, functions of heterocyst. So first in this title, identifications of heterocyst. Introduction. Theurus in 1884 was the first to report the cells in blue-green algae, especially in order to Nostococcus and Cyclinometals, except Oxylodendroceae family. Heterocysts are found in many species of filamentous blue-green algae. They are cells of slightly larger size and with more thickened wall than the vegetative cells. Identification. They are usually larger than vegetative cells, develop thick tumentary layers and intracellular hyaline button at the point of attachment to the vegetative cell, displaying a pale reduced autofluorescence. Such as heterocysts are easy to recognize under the microscope. Definition. Heterocysts are specialized nitrogen fixing cell formed filamentous cyanobacteria during periods of nitrogen starvation. Heterocyst is a differentiated cyanobacteria cells that carries out nitrogen fixation. Nitrogen fixation is a conversion of atmospheric nitrogen to ammonia in order to build organic molecules. It contains nitrogen fixing enzymes nitrogenase. Example of cooperations, heterocysts and photosynthetic cells in same filament exchange metabolic products, which is inactivated by oxygen. So heterocysts create an anaerobic environment by making 3 extracellular walls, degrading photosystem, producing oxygen, scavenging species. Heterocyst types. They have evolved multiple specialist cell types, including nitrogen fixing heterocysts, spore-like agonites, and the cell of multiple homogeneo filamentous of this. The development of heterocysts in the filamentous cyanobacterium hanabena species. Positions of heterocysts. Heterocysts usually occur singly in same species in pair and rarely in chain. When they occur singly, they are terminal or intractally in position. Lateral heterocysts are found in Nostococcus. Structures of heterocysts. Heterocysts are pale, yellow and barrel-shaped structure. These are covered by envelope and cell wall. Envelope of heterocyst is made of 3 layers: outer loosely fibrous layer, middle homogeneous layer, and inner lamellated layer. Middle and inner layer of uniform thickness except at the pole where they are more thicker. All the layer of envelope surrounds the heterocyst except at the junctions of the heterocyst with vegetative cell. The cell wall of the heterocyst is made up of 4 layers of vegetative cells. Cytoplasm include absence of carnitinoids and little amount of chlorophyll, but phylophills are absent. Photosynthesis is absent. DNA is present in the cytoplasm. All other granules and gas valules are also absent. At the junctions between heterocyst and vegetative cells, there is deep construction. Development of heterocyst. Any vegetative cell divides and forms two daughter cells. One or both the cells may behave protoheterocyst. Protoheterocyst enlarger in size secretes a multi-layer envelope out it sits in the cell wall. The pores appear at one or both the ends. It's a manner heterocyst pores become blacked with refractive material. They are called the polar granules. Now cytoplasm connections with adjacent cells is broken. Factors affecting development of heterocyst. Blue-green algae light inhibits whereas white and red light supporters. Concentrations of phosphate salts stimulate heterocyst formation. Absence of magnesium and iron inhibit heterocyst formation. Absence of combined nitrogens also have inhibit of effects. pH ratio also play an important role in inducing heterocyst form. Functions of heterocyst. Storehouse of reserve food materials. Sites of nitrogen fixation. Uses of filamented breakage play a role in sporulation. Vestigial reproductive structure. Produce substance which stimulates cell divisions and growth. What is nitrogen fixation? Nitrogen fixation is a process that implies the transmissions of the relatively non-reactive atmospheric nitrogen into its more reactive components. Why is nitrogen fixation important? Such reactive forms are suitable for crabs and support their growth. On the contrary, nitrogen deficiency stunts crab growth and healthily develop. About 90% of natural nitrogen fixation on our plant is biotic and occurs plant to soil microorganisms. Abiotic natural increases are lightning and UV rays. Alternately, nitrogen can be fixed with electrical equipment or industrial nitrogen fixing plant, and nitrogen fixing crops is a natural way to provide to plant adjust nitrogen without any industrial harm to natural. Using them in crop radiations allows nitrogen fixing of succeeding plant. Another successful practice is to use nitrogen fixing plant species in intercropping. Advantage of nitrogen fixing with cover crop. Legumes are known as the best nitrogen fixing plants and can be grown either as cash or cover crop. Nitrogen fixing cover crops breeding multiple benefits to farmers. Participate in nitrogen protect soil from aerations by covering its holding in place with a strong root system. Improve soil fertility when used as green manure. Retain soil moisture. Help in weed management with crop resistance. Serve as forage and grazing material for poultry and cattle. Attract pollinatory at the crop flowering time. Nitrogen fixing bacteria. As its name suggests, nitrogen fixing bacteria participate in the process of this nutrient fixation. Nitrogen fixing bacteria examples compare Rhizobium, Frankia, Aspergillus, and etc. Nitrogen fixing bacteria important to plants. The role of nitrogen fixing bacteria is to supply plants with the vital nutrient that they cannot obtain from the air themselves. Nitrogen fixing microorganisms what crops cannot get assimilative nitrogen for them. Bacteria take it from the air as a gas and release it to the soil, primarily to as ammonia. It is the only suitable options for plants because they can consume nitrogen only from the soil and only as nitrogen's inorganic component, which explains the importance of nitrogen fixation. So thank you for watching this video. This is a heterocyst and its importance of nitrogen fixation.